Now we just get to the let Professor John to present for the plenary talk.
Good morning, everyone. I'm delighted to be here to chair this first session. I'm John Marsh of the University of Glasgow. Ni hao da jia, wo hin gao xing zai jia. Okay, so it's my pleasure to introduce our first speaker, who's giving a plenary lecture, Professor Robert Prevedel from the European Molecular Biology Laboratory in Heidelberg, Germany, and he's going to talk to us on looking deeper with microscopy, pushing the optical imaging frontier in biology. Please.
Yeah, thank you very much for the kind introduction. I'm trying to share my screen and hope that it will work out. So can you see my screen?
Yes, you can see.
Yes, and I also see my laser pointer. Okay, well then, thank you very much again for inviting me to speak here at this wonderful workshop. I very much look forward hearing about the exciting science from all the participating research lab and hopefully engaging in some discussions and getting to know many of you in person. So today I'd like to tell you about what my lab research efforts are in trying to pushing the optical imaging frontier in biology, and in sort of what new optical imaging technologies we are developing in this respect, especially with regards to non-invasive imaging that enables to study biological processes in the context of its physiological and natural environment, ideally in vivo. And so I'd like to start this workshop with an inspirational quote by Freeman Dyson, who has said that the great advances in science usually result from new tools rather than from new doctrines. And I think this is very much true, because if we think about it, technology development is, as I see it, an important prerequisite for doing excellent research in the life sciences. And so with this, I hope you will also then agree and appreciate that microscopes are quintessential tools for the life sciences, because they allow us to visualize with molecular specificity various subcellular processes, and thereby come to a mechanistic understanding of how complex living systems operate. So in this sense, we often say seeing is believing, as microscopes allow us to learn from direct observation. So my lab develops different types of microscopes, which in these cases are all tailored to new, to provide new imaging capabilities in the areas of cell biology, developmental biology, and neuroscience. And so I work at the so-called EMBL, the European Molecular Biology Laboratory, and while 90% of the research here is on biology, we also have a history in developing various microscopy technologies, from visualizing single molecules in biological samples, for example, using cryo electron microscopy. Here, Jacques Dubuchet got the Nobel Prize just a few years ago for work done at EMBL, up through across all the length scales to imaging on the tissue and organism scale, which is one of my specialities. Okay, so with this little introduction now, I want to now highlight and the following and bring your attention to the omnipresent and recurring challenges that we face in bioimaging, where it is basically essential to capture and visualize both subcellular type processes, as you see here, that are happening in a mouse embryo during cell division, but also on an entire organismal tissue length scale, as you can see here in the Drosophila, the fly embryo. And this involves, more than 3 orders of magnitudes, both in the spatial as well as temporal domain. And it's very important for us to develop technologies that can capture these vast spatial and temporal scales in biology. And so the problem here is often that we want to visualize an entire embryo. Here, this is on the order of a millimeter, maybe. But we also want to do this in a very fast manner. And so what normally is done in microscopy, as many of you know, is that we employ, for example, a confocal or a 2-photon microscope essentially a point scanner. And then we scan the sample in 2 dimensions, then in 3 dimensions to make up our volume. But this is very slow, and it leads to this common trade-offs between imaging speed, the resolution at which we image, and the field of view that we can capture. So it's quite clear that conceptually new approaches are needed to overcome this challenge. And besides just the speed issue, there's also an additional concern with the single point scanners, and this is photo damage, which is very obvious in the case of confocal microscopy, where we focus the light to a single diffraction limited point and then scan this beam across our sample. We are subjecting the entire animal to a lot of phototoxic light. And so both the problem of the slow speed that I just mentioned and the phototoxicity were solved with the introduction of what is now widely known as light sheet microscopy. And so by introducing an orthogonal illumination or excitation beam here, we are restricting the phototoxic light to a single plane, while we can record all the fluorescence in parallel by an area detector, basically a camera. And this tremendously increased not only the imaging time, let's say for an entire Drosophila embryo, by at least 3 orders of magnitude, but it also reduced the phototoxicity considerably. And this really led to a revolution in bioimaging. And just to highlight 2 examples, here on the left, you can see work from Genelia, the Arendt's lab, where they were able, with light sheet microscopy, then for the first time to visualize brain activity in living zebrafish. Or here on the right, work by the Huisken group, which visualized the zebrafish heartbeat. But both of these works, for example, here, although they were seminal in the field, also had disadvantages. For example, the entire zebrafish brain could only be imaged at maybe 1 hertz or 2 hertz. So it was restricted to imaging calcium activity, while the dynamics of this beating heart were not acquired live, so not in real time, but actually reconstructed post hoc from different imaging planes. So while light sheet microscopy, allows us to not scan in 2 dimensions anymore, we still have to scan in the Z dimension, right? So how can we even sort of speed up that further? How can we even remove any scanning? And this challenge basically led us to the concept of light field microscopy, which is really a conceptually new approach of how you image 3-dimensional samples at high speed. And so the light field is basically a 4-dimensional function that describes the amount of light that travels in every direction through every point in space and can basically be defined by 4 parameters. So 2 regarding the location and 2 regarding basically the angle at which the light is traveling. And also, as you can hopefully appreciate, this 4-dimensional light field contains enough information to reconstruct a 3-dimensional scene or a 3-dimensional object. But now the question, of course, is how can we image a 4-dimensional light field with a simple 2-dimensional camera sensor? And to illustrate how this basic principle works in light field imaging, consider this camera here, this sort of pop-up camera, where we have individual lenslets here, 9. And if we take a selfie of our face, you'll see that, this image here now, this face is being imaged from slightly different directions. And so the number of perspectives here is, of course, the number of lenslets. And the lateral resolution is, of course, connected to the number of pixels behind each lenslet. But still, this sort of now allows us to reconstruct the 3-dimensional scene. So the advantage of doing this light field-based imaging is really that there's no scanning or mechanically moving part. And it's also quite simple and easy to implement, as you'll see on the following slide. So when we built one of these first light field microscopes, we got a so-called microlens array. So this is arrays of lenslets, around 100 times 100, so 10,000 of those. And we added this to the detection path of our microscope. And this allowed us to capture light field point spread functions that we can then deconvolve into 3-dimensional images. Now, I'm not going into all the technicalities, but I'm going to show you sort of the work that we've done here again in the area of zebrafish larvae brain imaging. And as you can see here, this is also calcium imaging of brain activity. And here we could do this easily at the speed of 20 hertz, so more than a factor 10 higher than what is was previously possible with light sheet microscopy, and doing this over quite large field of use. So that really was sort of a breakthrough back then, and it's led now to a flourishing field of light field microscopy in biology. And over the years, this has been now established in a number of different model organisms, from C. elegans, I just showed you zebrafish larvae, to the Drosophila brain, and even into the mouse brain. And so in case any one of you is interested in sort of further exploring this, we even have a website where we detail how to build a very simple light field microscope. Well, this is all great. So we overcame the challenge of how to image fast in 3 dimensions. But, you know, if in light field microscopy, of course, there are many advantages, but also a number of drawbacks that are inherent to the technique and that are maybe not often mentioned so much in detail. But sort of after a few years of working on it, I realized that some of the drawbacks were certainly due to the rather poor optical resolution, because we are trading off, you know, this the acquisition of this 4-dimensional light field. There has to be some trade-offs, so mostly it's due to the resolution, and especially the set resolution is usually pretty bad. We also experience so-called artifacts plane close to the native focal plane of the microscope, where we cannot reconstruct objects with high quality. And we are very sensitive to out-of-volume light. So if you, for example, excite an entire region, as you depicted here, but you only try to reconstruct a smaller volume, you will be basically impeded by the fluorescence that is emanating from the sample above and below. And this is quite obvious here in this case, again, in the zebrafish brain. So a few years back, we again set out to address the shortcomings, and we did so by combining the best of the 2 techniques that I just presented, basically light sheet microscopy and light field microscopy. So we use light sheet illumination to, again, you know, restrict the excitation of the light to the volume of interest, so that we do not excite any fluorescence outside our volumes. And we also incorporated a so-called multi-view detection approach in order to increase the resolution. And then if you see, this is so-called a 4-lens BIM system, for lens light sheet. If we have 2 direct detection objectives here, and these are looking at the sample from 2 different directions, these are the point spread functions. But if we do so from 2 90-degree separate views, then we can basically deconvolve these 2 views in order to get an isotropic point spread function, therefore increase the axial resolution of our microscope. So this is a simple schematic of the microscope that we then built. So it's based on 3 mutually orthogonal objectives, 1 for excitation and 2 for detection. And this now allowed us to really increase the resolution of the light field microscopy considerably. And again, here we are looking at a fish. In this case, it's a Medaka fish. This is in collaboration with the Whitbread lab here in Heidelberg. And we are trying to visualize the heart beating dynamics. And here what you can see is on the upper row here, this is a single view traditional light field microscope. And here's what you get by doing this with a dual view imaging. As you can appreciate, especially here on the right, while on the top in the single view, we can't really distinguish the different cell nuclei of this of the myocardium, so basically the heart wall. We can do this now very nicely in the dual view approach. And we are doing this by not sort of sacrificing any imaging speed, so we can still image this volume at more than 150 Hertz, which is sufficiently fast, as you can see here, to visualize the entire 3D heart as it's beating. And so we overcame the limitation that I mentioned earlier, where previous work had to reconstruct the heartbeat. We can now also, because we can image the heart in a single shot, we don't have to reconstruct from separate heartbeats. We can now also image, for example, individual red blood cells as they flow through or being pumped through the 2 chambers here in the heart. And what we can actually see is that some of the cells are not being pumped through immediately. They sort of hang out for a short while before they're being pumped out. And it's something that previously was not able to be resolved or actually being captured or visualized at all. Now again, so in this work, we overcame a problem, you know, in light field microscopy, in the sense that we significantly increased the resolution, but still there's an intrinsic other problem in light field microscopy, which is that it's a computational method. So for every image that we obtain, we have to go through an iterative deconvolution process to basically get the 3D volume. And this is quite computationally intensive. And if you imagine, if we make a movie out of thousands, 10,000s of frames, this can actually take a significant time. So very recently, then we turned to artificial intelligence in collaboration with another lab here at EMBL. And we designed a neural network that would replace the iterative deconvolution. And by doing so, we were actually able to reconstruct the line fit volumes by a factor 1,000 times faster than we've previously done. So that significantly speeds up the overall usability of the technique. And as you remember, we also had a light sheet microscope, in the last version. Now we can also use this to concurrently image with a light sheet our beating heart here. So you see in cyan here, so in blue, you can see the light sheet data that we get, and we can use that data to validate our artificial intelligence network predictions while we are imaging. So we have sort of a more or less a ground truth validation that is sort of inbuilt into our microscope. And that now really allows us to validate the predictions of the network. And also because we are now really fast, we can also basically do our visualizations live. We don't have to reconstruct them later on the computer. Okay, so this was the sort of the first part of my talk, in which I wanted to show you the work that we've done in our lab to really increase volumetric imaging, really fast 3D imaging. And I want to turn your attention to another problem in biology, or better neuroscience. And this is the quest to basically understand how our brains operate. And again, here I want to start with a quote, in this case from Barack Obama, who has said at the start of the Brain Initiative, that as humans, we can identify galaxies light years away, and we can study particles smaller than an atom, yet we haven't unlocked the mystery of the 3 pounds of matter that sits between our ears. And this is very much true, right? Like, while we understand how the universe works on all length scales, we understand very little about how the brain, for example, encodes information, how it computes information, and how this complex neural dynamics essentially lead to behavior. And coming back to Barack Obama's quote, of course, in physics, we have very powerful technologies, such as the Hubble Space Telescope or the Hadron Colliders, that allow us to peer into the galaxies and into the subatomic scale. And these are powerful tools to, of course, allow us to study how the universe works on all length scales. But we don't have a similar technique for brain neurobiology, let's say, for brain science. We need a similar technique that can capture different scales. And so now in the next couple of slides, I want to show you the work we've done in trying to push the scales in 3-dimensional optical recording of neural function. And so we've started this almost 10 years ago by working with the model organism C. elegans, visualizing the entire brain activity in those small nematodes. We've then sort of went on to image in the zebrafish brain. This is work that I basically just showed to you. But most recently, we're really interested in pushing the technologies in the area of mouse neuroscience. And of course, imaging in the mouse brain is a much harder challenge because the mouse brain is highly scattering. And so because it's a highly scattering, we basically have to come back to this point scanning microscopy approach. And again, now we have the same problem as I previously told you. We have to scan in 3 dimensions. So now how can we speed up that process with having the limitations of the mouse brain in mind? And the solution that we found a couple of years ago was basically that if we want to image a neuron, we don't need to interrogate it optically by diffraction limited scanning, because that way we basically get hundreds of points on a single neuron. Actually, it's much better to sculpt or shape the point spread function in the sense that we only interrogate a neural cell body by a couple of laser pulses. This is sort of the optimal sampling that can be achieved. And we do this by a process called temporal focusing, which is borrowed from ultra-fast laser physics. And again, I'm not going into the technical details here, but I just want to show you what it allows. And basically, this approach now allowed us to really go to almost an entire cortical column more than 500 microns on each side. And now we can visualize the brain activity of more than thousands of neurons that are engaged within this cortical region. Now, more recently, we've been working on custom behavioral sequencing analysis. So basically computational image analysis pipelines that are based on advanced features descriptions and dimensionality reduction to also track the behavior of the mouse and to not only reliably detect it, but also classify the behavioral output. In this case, this is about the face expressions of the mouse. And by combining this with calcium imaging now, we can correlate the activity of the motor cortex that is directly connected to the brain, to the facial areas. And we can correlate the brain activity to these behavioral sequences, for example, whisker movements, slight movements of the mouth and snout area. And this is now a very powerful technique for us to correlate the motor cortex dynamics to facial sequences and find the neural code that is used by the motor cortex or the brain to engage in complex behavior. Now, I told you the mouse brain is highly scattering, and this is actually one of the biggest challenges that we have faced nowadays in microscopy. Because if we want to image an entire brain, we have to somehow combat the scattering. And this is very much like, as you see on the top, very much like trying to drive in heavy fog. You know, the problem is that when we try to increase the light intensity, we just get more backscattered light and we're completely blinded. Now, it's very much the same situation is true when we try to image deep into the living mouse brain, which is also scattering. So this is a cleared mouse brain section, but just to highlight basically that when we try to image into the mouse cortex, the deeper we go, the more background we get. And this is sort of what is limiting us when trying to go deeper. But for a lot of neuroscience studies, actually what is needed is this superb, really sub-micron resolution to visualize individual dendrites and synapses. So we really need a very high resolution, but also at a very high tissue depth. So how can we approach that challenge? So for this, we started to work on 3-photon microscopy. And here, this is based basically on the fact that the attenuation in brain tissue is largest at around 2 different wavelength regimes, around 1300 and 1700 nanometers, which overlaps nicely with the 3-photon excitation of green and red fluorophores. So what we are doing in our work is really to push from this normal area of 800, 900 nanometers up into the near infrared in our microscopy. And this has a clear advantage for imaging deep, as you can see here. But just by shifting the excitation, we can already look much deeper into the brain. But by doing a 3-photon excitation of our molecules of fluorophores, we also gain by the higher nonlinearity. And this higher nonlinearity basically gives us less background signal and higher signal to noise, as you can see in this respect here. Because what is really limiting us in going deeper is the background fluorescence that is excited at very high layers in the brain. But however, you know, it's not always about just getting very deep into the brain. As I said, we also want to maintain very good optical resolution. And this is a big challenge, right? Because as we all know in optics, there's multiple things that can lead to optical aberrations. And in the area of, let's say, brain imaging, can be related to the microscope itself, or to these interfaces here, or just the inhomogeneities of the brain itself. But luckily, you know, we've learned a whole deal from the area of astronomy, again, physics and astronomy, basically, in how to correct these wavefront aberrations by using adaptive optics techniques. And this now really allows us to pre-compensate these aberrated wavefronts and to achieve the smallest, tightest focus inside the brain. And this is important because if we come back to this problem of visualizing single dendrites and spines on these dendrites, they are on the area of, you know, maybe a micron or even below a micron. And now if we have an enlarged point spread function because of aberrations, then we get only very little signal from these spines. But if we use adaptive optics to restrict the fluorescence, the excitation to the smallest possible point spread function, we also get a lot more signal. So we get higher signal and we get sharper images because the point spread functions are smaller. And so again, to sort of approach this problem in microscopy, you know that there's always trade-offs between these different parameters. Our goal was really to get deeper into the brain by using 3-photon excitation to enhance the resolution with adaptive optics, all while trying to ensure that the intravital imaging that we do leads to the lowest possible light exposure and corrects also for other problems in live imaging such as motion. And so we've built a 3-photon microscope that incorporates these deformable mirrors, and that also incorporates a fast FPGA that measures the electrocardiogram signal from the mouse and basically synchronizes this to the microscope so that any motion would not be visible in our images. And this now really allowed us to image deep into the brain. So in a normal 2-photon microscope, we can only image to around 600 micrometers. But now, as you can see, with our microscope, we can go deep below 1 millimeter and deep into the hippocampus. We can also show that adaptive optics really leads to an improvement, not only in signal, but also in resolution, as you can see here on these 2 slides. Now, when we zoom in, you can also appreciate here, this is the dendrite. It's fairly almost not visible without AO, but with AO now we can see the dendrite and individual synapses. So that really makes a big difference. We can also show the same capability even at larger tissue depth here in the hippocampus, below 1 millimeter in the brain, where again we get a very high signal enhancement, not only from the somatous, but especially from the dendrites. Because remember, the smaller the structure, the higher the signal enhancement that we would expect. And this now really allowed us with adaptive optics to push the imaging depth to unprecedented depth. Here, for example, you can see that we are imaging deep into the hippocampus, which starts at around 900 and goes all the way to 1.5 millimeters. And here with adaptive optics, we can now visualize nicely these dendrites. And this is really the deepest adaptive optics demonstration to date that has been achieved. Now, this is structural imaging. Of course, we also wanted to show that we can do functional images. And for this, we collaborated with a colleague here in Heidelberg, Amit Agarwal, to visualize astrocyte calcium signaling in vivo. And so the problem that Amit had is that the brain is composed of many different astrocyte types, especially fibrous astrocytes that reside in very deep brain regions. So they are not in upper regions, only in deeper brain regions. And the problem here is that with 2-photon microscopy, people were restricted to just protoplasmic astrocytes, and fibrous astrocytes had not been imaged. So what we could show now is that with our imaging technique, we could very nicely visualize the calcium activity of different astrocytes. Here, protoplasmic astrocytes, very close to the corpus callosum. Near on the right, you can see how an automated imaging pipeline basically reads out all the different microdomains of these astrocytes. Now we can also show that adaptive optics is crucial in basically enhancing the signal. As you can see here, adaptive optics is off. We can barely see any signal from astrocytes, but as we switch adaptive optics on, now you can see that we can clearly resolve these dynamics. And we can plot the difference. And what you can see here is basically that adaptive optics leads to a much higher, but they're really the advantages that they have 3 orders of magnitude higher 3-photon cross-section than normal fluorescent proteins. And to really show or demonstrate the capability of these new nanoparticles, we did joint work in which we injected these nanoparticles into the vasculature. And indeed, we can use this to image it to very high brain depth, down to around 1.4 millimeters. So this is really excellent work, and we are currently also investing more efforts in order to use nanoparticles to visualize brain function, but also brain structure. Now, the problem very often that we have is that, as you see, we need to label stuff in biology. Sometimes we cannot label our tissue structures of interest. And this is why lab is also working in the area of optical coherence tomography, which is really a nice modality to visualize structural information in a label-free manner. And here I'm very happy that a very talented research scientist as part of my team, Ling Wang, who's really pushing these developments in our lab. So the working principle of optical coherence tomography, just for those who are not aware, is a little bit similar to ultrasound. It's just sort of an optical ultrasound technique in which we send ultra-short laser pulses into our tissue samples, and whenever there are refractive index inhomogeneities, we get slight back reflections from the different layers, which we can then detect through an interferometric approach. And this allows us here, for example, in the mouse, to image through the skin and to visualize small tumor masses non-invasively at the very high imaging depth. So more recently, Ling has built a fantastic dual view OCM microscope, so optical coherence microscope, to really push the resolution and to again enable the visualization of fairly large structures in biology. And here the advantage really is that here we achieve a more or less isotropic resolution down to around 2 microns in a very fast modality. So Ling has built this microscope with the goal to visualize entire organisms on behaviorally relevant timescales. And so here we work with the lab of Isam Ikmi, for example, at EMBL to visualize the different tissue layers in Nematostellar vectensis, which is a small sea creature that can grow to the size of 1 or 2 millimeters. And as you can see on the right, OCM allows us to distinguish the different tissue layers. But we can also use the microscope and the fast speed to visualize the behavioral dynamics of this big sea anemone. So here again, this is on the order of a millimeter. And as you can see, the microscope is sufficiently fast to image the entire volume in a behaviorally relevant time scale. But we can not only do this with the sea anemone Nematostella, we can also, for example, now use this to visualize the embryogenesis of the Drosophila larva. And as you can see here, you can see all the different tissue folding and invagination events. So it's now really for us an important imaging modality that we want to push further in the future and show that it's also useful for developmental biology purposes. Now, but again, I told you the big grand challenge in microscopy is to image very deep into scattering tissues. Now, optical coherence tomography is still an optical technique. So how can we basically try to image deeper? And for this, we are starting work here in the realms of photoacoustics. And this is motivated by the fact that with optical techniques, we get very high spatial resolution, but we are restricted to maybe a millimeter of depth in the mouse brain. So at some point, if we want to image deeper, we have to give up light, which is strongly scattered by tissue, and instead turn to longer frequencies such as sound. And so for this, we exploit tools such as photoacoustic imaging, where the tissue is diffusely irradiated by light, which is then subsequently absorbed and which leads then to the emission of acoustic waves. Now, these acoustic waves are not scattered and therefore can be detected very well. In our lab, we are relying on an optical technique to detect these acoustic waves. Basically, we are using so-called Farié-Perot etalons. These are self-built small etalons that can sense the acoustic vibrations and translate the acoustic signals into a light-based signal that we can then detect. And this allows us, again, non-invasively and in a label-free manner to, for example, visualize the vasculature here inside the living mouse. As you can see here, we achieve fairly good resolution down to around 50 microns up to around a depth of 5 millimeters. And that now allows us to, for example, look at tumor regenesis, or the progression of tumors inside the mouse over several days. We can image the same regions and see how this melanoma tumor cells that express melanin, which is very well absorbed, how this sort of grows over time. All right, so I think I have 10 more minutes or so, and with this I wanted to again sort of turn your attention to a different problem in microscopy, or in biology actually. And this is what I call the dark matter of modern biology. And I call this the dark matter because a lot of efforts in biology these days are related into finding the genes or the chemical signaling that lead to biological behavior. And this, all these genes and chemical signaling can very easily be labeled by fluorophores. However, there's also, of course, a physical dimension to biology, which cannot be labeled by fluorophores. And by now, we know that physical forces such as, membrane tension or cell-cell adhesion or mechanical properties of, single cells and tissues are very important also for biological processes, such as, for example, in stem cell differentiation. So when a stem cell decides to differentiate, in different cell types, such as neurons, muscles, or bones, it actually uses the environmental cue, the mechanical cue of the environment. It has been shown that depending on the substrate stiffness, this stem cell would develop into a different cell type. So this is basically just to highlight and motivate why, physical forces and mechanical properties do matter in biology. But the problem that we now again have in biology is that we don't have the ideal tools to measure those mechanical properties in a non-invasive fashion. And this is because, in a lot of biomechanical studies, either atomic force microscopy or micropipette aspiration are being used. But both of them rely on external loading, so they push or pull on your sample. And by sort of this external mechanical loading, of course, we are interfering with the biology. We are perturbing the sample, but we also don't really get any information from the inside of the structure, right? So with AFM, we can probe the surfaces, but we cannot go inside our samples. Now, of course, we also have optical techniques such as optical elastography that somehow overcomes this limitation, that can sort of measure also inside tissues, but it currently has a very low spatial resolution, certainly not on the single cell level. And there are other techniques such as injecting particles into the tissue to see how the stresses act on these particles, but this is quite invasive. So overall, there are many drawbacks to the current biomechanical tools that biophysicists have, in the sense that they are non-invasive, they are low resolution, and they mostly work only on the surface. So we cannot probe inside. And now, so in the remaining 10 minutes or so, I want to show you how we are using a novel optical approach called brilliant microscopy to basically visualize, like we are used to in microscopy, to visualize these mechanical properties inside living cells and tissues, and thereby overcoming all the previous mentioned limitations, such as being non-invasive. We have high resolution because with light we can focus to a very small point, and we can, of course, we are getting 3D access to our sample because we can easily focus inside our cells and tissues. Now, how does brilliant microscopy work? Well, the general principle is based on the phenomenon of brilliant light scattering, which is the interaction of light with thermally induced subtle material vibrations called phonons. So because we are, like any biological matter is basically at room temperature, there's tiny, vibrations inside our sample, which are so-called acoustic phonons. And these phonons can exchange energy with the light when they're being scattered. And so as a large proportion of the light is just backscattered without exchanging energy, this is the so-called Rayleigh scattered light, but a small portion of the light can be scattered and exchanges energy. This is the brilliant scattered light. And from the spectrum of this brilliant scattered light, so here you can see the Rayleigh, here the brilliant, Stokes and anti-Stokes, whether energy is given or taken away from the sample. From this spectrum, we can learn a whole lot about the mechanical properties of the sample. This is because the so-called brilliant shift, so the energy shift, depends critically on the sound velocity of our sample, as you can see in this relation here. And so by basically measuring the brilliant shift and the line width, we can then calculate what the stiffness and viscosity are of our sample. So brilliant microscopy is really an optical technique to both sense the elastic component as well as the fluid component of a biological sample. And as we all know, cells and tissues are composed of both things, fluid and solid components. Now, in order to establish brilliant microscopy in biology, we built a confocal version of a brilliant microscope. And here again, this is just a schematic picture where we excite our sample, and the brilliant scattered light travels through two highly dispersive optical elements, so-called vipers, that separate the brilliant scattered light from the predominantly Rayleigh scattered light here. So these two peaks here are the brilliant scattered light. And now the advantage here really is that with this brilliant microscope, we can achieve a very high optical resolution. We can probe really with sub-micron precision. But the two Viper devices here also allow us to image the spectrum or detect the spectrum with very high accuracy down to below 10 megahertz. So this is on the femtometer scale. And so we built a spectrometer and coupled it to a normal research-grade fluorescence microscope. And now this allowed us to investigate the mechanical properties of, for example, single cells. Here we can see subcellular components, here the nuclear envelope, the nucleolus, here in this mouse embryonic stem cell. But we can also probe the mechanics of larger tissues. And here, for example, you can see the muscles and vacuoles of this zebrafish larvae tail. And zooming here into this region here, it's quite interesting that we can even make out a small extracellular matrix layer here that separates the notochord from the inside. And this extracellular matrix layer has been suggested to be a very stiff matrix, because essentially it allows the organ or the tail to grow in a one-dimensional direction. So these vacuoles are being pushed to the back, and this allows the animal to grow in this dimension. So then we need a high rigidity here in this case in order to constrain this development. And so indeed, in our spectrum, we could see the differences between the outside tissue and the small extracellular matrix, and we could measure it with very high precision. So, and these are sort of just some highlights of our work that we've done recently, in which we want to show that, brilliant microscopy really is a tool that can record with high resolution the mechanical maps of cells and tissues. Okay, now this is of course still looking at very small samples, but also in brilliant microscopy we have a similar problem as we are having in other modalities, where we can't really image an entire sample at a very high speed. And this is why more recently we've been working on a new brilliant microscopy platform that allows us to image biological processes in 4 dimensions. In our particular case here, we built a platform that is compatible with embryo culture techniques that then allows us to image embryo development over time. And here on the right, you can see this is a mouse embryo. Here on the left, this is fluorescence imaging, and here on the right, this is the brilliant imaging that we can now correlate, and you can track every cell and measure its mechanical properties in 4 dimensions. We can also do that in other model organisms, such as, again, here the fruit fly. As you can see here, this is the embryogenesis of the fruit fly, and as you can see in a moment, there are very strong tissue invagination.
Excuse me.
Yep.
We see another the notes. Can you check your screen?
Okay. Now, better?
Yes.
Okay, sorry about this. So maybe I go back a few slides. Did you see this?
We see this
Yeah,
one,
so-
yes.
Okay.
Yeah, we saw that
great.
one and then it started to go wrong.
Okay, sorry about that, yes. Yeah, so we are building this new imaging microscope based on brilliant scattering to really improve the speed of our technique. And here, this is maybe what you didn't see. This is the mouse embryo, and now we can sort of follow every single cell here in fluorescence and correlate it to the mechanical properties that we see in the brilliant channel. But then, of course, we can also do this in other model organisms. Here, this is the fruit fly, where you can see in a moment here this tissue invagination events, and they are also hypothesized to have a very strong mechanical component. And now we can also image this tissue regions here with very high speed. And as you can see here in a moment, when invagination starts, we get a very, we get a very strong upshift signal that you just saw here. That basically also shows us that mechanical properties are highly changing on a very fast time scale in these morphogenic events. And so we are building these devices really with the vision to sort of also revolutionize four-dimensional imaging of biological function in the future. And here really the vision is that we can also provide four-dimensional physical maps at high spatiotemporal resolution. In the sense that we want to also image the physical properties like forces, tension, and material properties in addition to genes and signaling molecules. And so that the key applications in this are, of course, that we hope from this to gain a better understanding of tissue morphogenesis in the native environment in which it's happening, but that we can also integrate all this mechanical information into previously assembled gene or structure maps, as has been also done here at EMBL, where neighboring labs have, for example, built an entire gene expression atlas and a single-cell morphology atlas of the developing Platinaris model organism. And so we hope to add mechanics to this picture. But we can also combine now the mechanical information with the different spatial omics effort that have been performed recently, which also allow to map gene expression patterns in a spatially resolved matter over time. So with this, I'm almost at the end of my talk. This is an image of EMBL at sunset. So it's a very nice place to work in. So if you have any chances, please come by to visit once the pandemic at least is over. And I want to basically end with this slide, which shows the different microscopy techniques that I've been talking to you about today. And just sort of to summarize, we work a lot in high-speed 3D imaging, both using light sculpting, a 2-photon technique to look at brain function, light field microscopy in transparent animals to look at very rapid dynamic events in 3D. But then we also work a lot in deep tissue imaging, where we exploit 3-photon microscopy or photoacoustics to look again at brain function or tumor biology in a living mouse. And the last part was on brilliant microscopy, in which I showed you how we can use light or exploit the principle of brilliant scattering to detect mechanical properties of cells and tissues using light. And so with this, I want to thank my entire lab. This is a picture recently taken at EMBL. I want to thank all my funding and, of course, the many collaborators. And I'm happy to take any questions.
So thank you very much. Do we have questions from Glasgow, first of all? Yep, several.
Yeah, now it works, yes.
Excellent. So my question was, at a certain point, you were presenting this deep neural network method to reconstruct the image.
In light field microscopy, yes.
That was basically taking away the convolution method and replacing with deep nets, right?
Yes.
So my question was, could you please give us some details how you trained it and what kind of deep networks you trained, what was the input specifically and whatnot? Thank you.
Yeah, no, sure. So, we've used a very typical UNet structure, in case you're a little bit familiar with the different structures in deep neural networks. And of course, these need to be trained, and they need to be trained with, high resolution ground truth data. And so the way we trained it was basically, I only mentioned it briefly, we have this concurrent light sheet modality in our microscope. So this scans through our sample and provides us with very high resolution individual images, not volumes, but images. But then sort of we can train the network or validate the network with these high resolution images. So this is basically the input that we feed into the network in order then to, or basically not the input, that's sort of the validation layer that we use to compare the predictions of the network. And then if it's not great, then we sort of refine, and by this way the network is being trained. So it typically takes on the order of a few hours to train these networks. Maybe that's also an important thing to state here.
Okay, thank you. The question. I think you're going to come forward to use the microphone.
Talk here.
Oh, there's one there.
Yeah. Thank you for your excellent presentation. I have one question regarding your last brilliant microscopy. The first one, does the surrounding tissue affect the measurement? So that is the first question. Second question, you have mentioned that the mainstream current method like AFM has a lot of problems, but most of people use it. How do you envisage that people will take the brilliant microscopy, this approach?
Okay, yeah, so maybe towards the first question. So what we measure is sort of everything.
The first question is, does the surrounding tissue affect the measurement?
Yes. So by surrounding tissue, of course, it depends on, sort of the scale. So our point spread function is like from a confocal microscope. So maybe a 500 nanometers wide, maybe a micrometer long, and everything that's within this volume, this is being measured. Yeah. So if like with the ECM layer, this is very thin, even just a few hundred nanometers. In that case, the sort of the surrounding tissue did affect the measurement, in the sense that, as you saw in the spectrum, the spectrum was consisting of basically two peaks, one from the outside and one from basically the extracellular matrix. So yes, the outside does influence the measurement if, we're looking at very small spatial scales. But basically, we are measuring everything that's within our point spread function. And so the second question regarding AFM, of course, I think brilliant microscopy has many advantages. I mentioned some of them, to be able to go into the sample, have high resolution. And it's an optical method, that can be coupled to a microscope. So my hope would be that people who are using currently an AFM that is very often coupled to a microscope already, that, one could also sort of think about multimodal studies, in which you combine an AFM with the brilliant microscope, and then depending on the question, sometimes AFM are more suited to the question, especially if you look at low frequency mechanics. And so what brilliant measures is sort of a high frequency mechanics, but it can be also very informative because it gives you a sort of an instantaneous measure of your mechanical property. So I'm not claiming brilliant microscopy can replace, all the other methods, but it addresses some of the weaknesses of those methods.
I don't know how to see other questions. There's somebody with their hand up, but I don't know how to see. I don't know who it is.
John, are you going to allow us some questions in the NGUSD?
Can you?
Sorry. Yes, I'm Xu Fang Liu. Hello, Robert.
Hello, hi.
Hi, long time no see. Very nice to have you to give this talk. You guys showed a very nice image of Ambo. How is the new building for this imaging center?
Yes, I can.
Complete?
Yes, so we have a new imaging center here at Amble. That's correct. Yes, it was completed last year, already during the pandemic, and it started operation very recently. So it's the idea, maybe for everyone who are interested. So the idea is it's an imaging center that allows to provide service, about the type of microscopes that we are developing. to the outside of EMBL. So worldwide, researchers can apply. They can apply for microscope time at this imaging center. And if the application is positive, then they can come with their samples and perform the imaging at the imaging center, assisted by very professional scientists that help mostly biologists, right? Then to get the data that they want from this new type of microscopes.
Right. Ambo obviously has put a lot of effort on this biological imaging that we all can see. I think you are in a very good position in the forefront of the world. Yes, it's very nice for you to brief us all your advancement. I thought just have a very small question. Obviously, you have done a lot of ways to develop a lot of ways for imaging. And yet you are in the most of your effort are on imaging things in vivo, in depth, in certain depths. Some of that come over 1 millimeter. But you are, when it comes to 1 millimeter or even deeper, you used a sort of 4-dimensional measurement. Is this 4-dimensional 2-angle and 2 Cartesian dimension? Are you eventually trying to get out to a 3D imaging or what?
Yes. I mean, so most of the imaging we do is in 3 spatial dimensions over time. So then I call it 4-dimensional imaging. I mean, as you said, we are working on separate methods. All of them targeted to different spatial and temporal timescales. For example, light field microscopy, very fast in 3D, but it can't go very deep. Photoacoustics, for example, can go very deep, but has lower spatial resolution. So we are sort of trying to approach the different problems in bioimaging from different angles.
Sorry, I have another question, just a small one. You obviously combined the elastic measurement and the balloon measurement together to combine them together to make a new type of imaging. Do you have any other thought of combining this multiple dimensional measurement with photoelasticity or other elastical or you call it dark matter, dark measurement? Do you have any other sort of other type of technological image technological combination?
Yeah, so that's a very good question. We are not actively pursuing any sort of other modalities at the moment, but I think it would be very exciting to, for example, especially see how force measurements can be integrated also with brilliant microscopy, because brilliant microscopy measures material property, stiffness, elasticity, but it can't measure forces. So I think it would be truly exciting to have both force and material measurements.
That's great, John. We have finished our question over this slide.
Thank you.
John, can you hear us?
Yes.
John, can anyone, when the people giving talk, can the people just give one word, short sentence of introduction to himself? Is the one question, is it Huabing Wang, Professor Wang? John, can you hear us?
No, clearly.
Oh gosh.
Sorry. Did you hear?
I just want people to introduce themselves when they asking questions.
Okay, yes. When they ask a question, yes. We'll ask people to introduce themselves in future. Okay, so thank you very much for an absolutely excellent presentation, and thank you for attending our conference.
Yeah, thank you very much.
So we now have a short break until 10:40 for coffee, at least in the UK. what time of day it is in China, but an appropriate refreshment. So,
Okay,
thank you.
So we have half an hour break.